Welcome back, everyone. Today we're going to be looking at changes in biodiversity over time. So this links to the topic that we did last week, and it is the second part to Unit 4, Area of Study 1. There are three key dot points that we're going to be covering under this topic. We're going to be looking at geological history, so the rise of multicellular organisms from unicellular organisms previously, so how we combine these multicellular organisms to create animals that live on the land. We're looking at plants, the ones that were flowering, so different forms of reproduction. We're looking at mammals and all different types of animals within this category as well. We're going to look at evidence for this change over time. So this is where we start to look at paleontology. We're going to look at the fossil record, and we're going to look at a few different ways that we are dating fossils. We're going to be looking at carbon dating and those types of things as well as a part of this topic. We're going to look at biogeography, so where things are and where things have been on planet Earth. Developmental biology, so looking at the development of embryos and how we can compare embryos in different animals and look for similarities and differences that prove how related things are to each other as well. And the last one is structural morphology, so looking at structures, so like our hand and the bones and things that are in that, and how they're similar and different to other organisms as well. And the last one that we're going to be looking at is different forms of evolution, so convergent and divergent evolution, as well as mass extinction and things that are causing different organisms to die off. So we will be talking a little bit about the dinosaurs today. What we need to start off with by doing is looking at the entire history of planet Earth. So about 4.6 billion years ago, that's what we propose the origin of the Earth to be. We break that down into lots of different eras, so different sections of time. And within the eras, we have periods. Within periods, we have epochs. And then within that, we have the different periods of time. So if you look at Homo sapiens, we're looking at about 0.2 million years ago that we're looking at the version of Homo sapiens that is us starting to come into existence. But we're going to look at a whole general period of time there as well in just a minute. There's lots of different things that have changed during this period of time. There are, of course, physical changes and structural changes to Earth, so where mountain ranges are, where water is, and those types of things. But also we're looking at climate changes. When we went through the Ice Age and the large proportion of Earth being covered in ice versus the temperatures that we're having now and the temperatures that we had back then. So those different temperatures, different climates, different physical attributes of Earth cause different selection pressures on organisms as well, and that's just something to keep in mind. We're going to be looking at studying rocks in terms of tracing these changes over time and the evidence that's within these rock features that prove the changes that have been occurring over time. The big ones that we're going to be looking at in terms of change is position of land masses, so continental drift, so Pangea, when all Earth masses and things were together, and how continental drift has separated them, and how as a part of that drift we're looking at creating subspecies in different areas but that have common ancestral links. Global temperatures and how those changes have impacted. Another one to consider is oxygen levels. We rely on a high amount of plants being on Earth to produce a lot of oxygen for us. And the last one is sea levels. So as our Earth temperature rises, more water will be melting, that's in our polar caps, which will cause the sea levels to rise, pushing more Earth underground. And that can change our water temperatures and all those types of things as well. So they're just a few little factors to keep in mind. All those changes affect vegetation, so plants, and where they live. So if you think about the rising sea levels, what was once above ground and can photosynthesize and get its carbon dioxide from the air, now is it underwater and isn't getting the same amount of oxygen as it needs to be able to get them out through the water. It isn't getting the same amount of sunlight as the light gets reflected and refracted because it's going through the water. So those types of things lead to changes in plants so that they're able to live underwater versus above water as well. So all these changes are very important when we talk about how they've changed over the course of the life of planet Earth. What we're going to do now is look at the last 600 million years. Down the bottom here we have different periods of time, and you'll notice that they're given names. So there's the Cambrian period, the Denovian period, all the way through to Jurassic, which should sound very familiar to you guys with Jurassic Park. And we're talking about the different life forms that were found during these periods of time. So if we go back to the nearly 600 million years ago, we're looking at the first arthropods and the first fish having evolved. Obviously, with time, those organisms are going to be changing. We've got the first land plants, which were between 400 and 500 million years ago. And the ones that you really need to be keeping in the forefront of your mind are the first flowering plants, which was between 100 and 200 million years ago, and then the introduction of things like bees about 100 million years ago to utilize those flowering plants and help them pollinate each other. Now we sit about here. We're part of the genus Homo, so we are Homo sapiens, and we are right at the very end of this little graph. So if we look at this chart, our time on Earth really is quite small and almost insignificant when you compare it to how long plants and bees and other mammals have been on planet Earth. It's these small changes that happen to how the Earth is structured, our temperature, the amount of oxygen that's been available for us, that encourage evolution to be happening. The small changes that happen but over a longer period of time is what creates these things to happen. So we're looking at for here that really in the grand scheme of the history of Earth, we don't really mean a great deal. We're quite insignificant. We're not a long-term survivor, I suppose you could say, but yet we're having this very large impact on where we live. We're changing it so much. We're changing the way that the Earth looks. And if you look at it in some of those pictures from space, you can really see the impacts that we're having here. So this is another version of the geological timescale. Very similar to what we were looking at before, except this one has more major events within it. So down the left-hand side here is the time period, and MYA is millions of years ago. And then in here, we're looking at how long that would be on a line. So today would be at the 0 centimeter mark. 100,000 years ago would be 0.1 of a millimeter, so not even 1 millimeter. 22 million years would be 2.2 centimeters. So that if you were to draw these things out and you're going to copy this into your book to look at making a timeline of these different events, just to keep that in mind. This column here has all the major events. So we've got the present. 100,000 years ago, we had the modern form of humans, or our Homo sapiens. 2.2 million years ago, we're looking at grasses. The first apes were 33 million years ago. And on the right-hand side here, we have different eras of time in terms of when these things were originating. And it's given some suggested colors there that you can be using in terms of categorizing these periods of time, just so that you are aware of that when you're copying this into your workbook. So I recommend hitting pause and copying this table into a timeline which should be very similar to what was just on the slides 2 seconds ago. Pause and copy this into a more linear form, which is another way that this could be coming up in your exam. Now, the geological timescale was developed in the 19th century when we're looking at rocks and we're seeing these common characteristics in rocks, but they're in different locations. Distinctive fossils are being found in them and all those types of things as well. So where all these things are found is generally one of the things that we use to be naming the groups of fossils and the groups of rocks that we're finding in those areas. So their distinctive nature helps us to name them and be able to tell them apart from each other. For convenience's sake, this is where we look at breaking the time scale into periods and eras. So it's a hierarchical interval. We're looking at breaking things down so that we can group similar occurrences together and see what happened in different places based on what was occurring. So if we look at this significant rise in O2 levels, we can look at its impact in different locations around the world. But we know that that roughly had to happen based on the appearance of eukaryotic cells and then more multicellular organisms becoming a result of that. So before we really see all these fossils, that's where we get into the Precambrian era, where we're really looking at not much evidence still existing of these things. You need to remember back to your Year 8 science where you looked at the rock cycle, and by now a significant amount of time has occurred. So any evidence that we would have had before this, or even, it's talking about being before Earth was formed potentially, but from the origins of Earth's first creation, they probably don't really exist too much anymore because of the factors that are affecting there, like temperature and pressure, and them getting further down towards the center of the Earth as well. So what we're going to look at now is paleontology, and in particular we're looking at the evidence that we can get from fossils around evolution. So a few things to keep in mind. Fossils aren't bones. Fossils are rocks. Fossils are evidence that something was there at a particular point in time, okay? So they are the preserved remains, and they don't always have to be whole body remains either. They can just be limbs, or it can be something that showed that an organism was around. Think footprints or feces. All of those types of things can be fossilized as well to have evidence that something existed in a particular location. Key term here, fossil record. All fossils that have been found, everything that is discovered, sits as a part of the fossil record, okay? And what we do is we track their position through the layers of the rock that you can see here on the side. Obviously, the further down that something is, the older that it is going to be, because new layers of rock have piled up above it over and over and over again. Now, this is where things can get a little bit confusing for you. Things that are further down are older, things that are closer are younger. So the pattern of evolution and change will go up. So from the old down here, up through the layers to the new. And this is where we get that time being new, but the time for evolution occurs through those things as well. So the closer you are to the top, the more evolved something is going to be to the version of it that's at a lower level down in terms of our rock layer. So we can compare the different layers, which in the science world we call them strata. We compare the different strata of rock, and we compare the fossils that are found in them to show that these organisms are slowly changing over time. And there are things that we can propose from that as well. So are they getting sharper teeth, which would show us that they're eating a different type of food? Is there evidence to show that they had more fur? Was it getting colder? So there's a whole range of different factors and evidence that we can be getting from these fossils around what the world was like at that time. How did they live? What factors are influencing their survival? There's a whole range of different stuff that we get just from looking at fossils. So relative dating is another way that we can compare them. So we spoke about the oldest fossils being down the bottom here and the newest ones being at the top. Now, when we're talking about relative dating, it is just that. It's relative. It's not an exact science. It's not perfect. We're not giving it an exact date and saying, hey, you're this many years old. Not how relative dating works. When we're talking about relative dating, it's comparing organisms that are in one layer to the organisms that are in another layer and saying, well, because you're above that, you must be younger than the one that is below you. So that's what we mean by relative. If you're further down, you must be older. Further to the top, it must be younger. So we go, oh, well, maybe you're about this old if we compare you to some of the other things that we know that are around you. But it's quite important that this is an undisturbed sequence. So if there's anything that's impacted on it, so say they've built something there and they've taken layers of dirt away, obviously the layers are not going to be complete, so the absolute dating there just won't be as accurate as it would be in a undisturbed area, okay? Look, there is stuff that we can get by using relative dating, but it's not perfect. So if it comes up in your exam, you need to keep in mind that it is just comparing different things in different locations, and we're not looking at getting an exact age. We're going to talk about carbon dating and some of the other options, which give us better information in terms of how old something is. But we can get things like a climate chronography, so roughly the climate changes that are occurring over time. We can use tree ring dating as a part of this. You might have heard the old wives' tale that if you chop down a tree and you look at all the circles inside of it, then you know how old that tree is. That's what they're talking about there when they talk about dendrochronology. Oh my gosh, I'm never going to be able to say that word right. But if you count the number of rings that are inside the layers of a tree, you can roughly tell how old that tree is. And you can look at factors that were affecting them roughly at that period of time. So the trees grow from the inside out and they're creating new layers that way. Another thing that we can look at doing is ice core sampling. This is where we drill down into where there would be like a glacier or a significant amount of ice. When you pull that ice core up, it's like a long tube of ice, and you can see a whole heap of different things within it. So say it melted and it got really dirty, and then it got really cold again, and now you got more ice on top of it. You can see all those types of climate changes when you're looking at core sampling. And again, that's just relative to comparing the ice that's at the top to the ice that's further down. The one that's further down must be older. And we spoke about stratigraphy just before, when we were comparing the strata and the lines in terms of looking at fossils that are found within them. If we want to be more specific, there are two types of radiometric dating that we can use. There's this carbon dating, and there's also potassium dating. Both of them have advantages and disadvantages because they are different elements that are found. This gives us an actual date for how old the fossil or the biological sample or that rock is going to be. So as we said, relative dating is just the lower down you are, the older you are, the higher you are, the younger you are. When we're looking at giving an actual date, we're looking at how many years something is old, okay? And we can look at, in this case, they're looking at dating volcanic ash, and they can see that there's several layers of volcanic ash in the strata in this particular location. So they take the volcanic ash, they do some tests on it, probably in this case it's going to be carbon dating, and they're looking at how the carbon isotopes change over time to determine the millions of years old that something has to be based on its half-life. Now, if you do chemistry, you would have already heard about a half-life of an isotope. For those of you who don't do chemistry, you don't need to know this in a great deal of detail, just what half-life actually means. The two other techniques that we can use to get an exact date for an organism is electron renaissance, which is a type of imaging that we use, and then there's luminescence techniques as well. So we're going to start to talk about a couple of these different things now. Okay, so if we focus on radioactive isotopes, what we're talking about here is the atoms that make up rocks and how they're decaying over time. So how they're changing and how the atoms within those rocks are changing over time. So we're looking at radioactive atoms, and they're generally quite unstable. So they'll start to decay or revert to their non-reactive form over time. So if you're reading an exam and it talks about radioactive atoms that are found within rocks, you need to keep in mind that it's over time that they're becoming non-radioactive, and it's that change that we're measuring when we talk about radioactive dating, carbon dating, or any of those types of things as well. We look at their half-life when we determine how things are decaying. So half of the unstable atoms will have decayed in their half-life. So if we look at over time, what's happening to them, we look at how long it takes for half of the atoms to go from being radioactive to being non-radioactive, and that's what they call the half-life. So they can calculate how long it takes for 50% of the atoms to change, and then from that we can look at extending that into a greater sample of atoms that we're finding within fossils and rocks and those types of things. So the options that you have in terms of your radioactive dating or radioactive isotope dating is uranium. Uranium turns into lead or some other elements there as well. But the big one that we tend to use most commonly is carbon, and carbon changes from carbon to nitrogen. And potassium is another big one for us as well. Potassium decays into argon. So we're looking at the decay of a radioactive atom into a non-radioactive atom. That's what radioactive isotope dating is. Let's dive into carbon-14 dating a little bit deeper here. So we use carbon-14 for a fossil that's relatively young, and it still has some organic material in there. So it still has some carbons and hydrogens and oxygens stuck together. So that's relatively young. It's not entirely a particular type of rock yet. It doesn't have any of those things happening. So when the living thing dies, we're looking at the radioactive carbon-14, and it decays into nitrogen-14. So they're two different elements. Carbon-14 is the radioactive one, and over time it's going to break down and decay into nitrogen-14. What we do is we look at the percentage of carbon-14 and nitrogen-14 and how they've changed over time to calculate that half-life. So it is accurate up to 500,000 years. So that's not really that long when you think about the millennia that we were talking about in terms of billions of years of life on Earth and those types of things. It's most commonly used for testing human-like fossils because, as we spoke about before, in the grand scheme of things, we haven't been around for very long. So 500,000 years is more than enough time to be able to use carbon dating, that carbon-14 dating, to determine how old a human fossil is, or things that were around at the same time as humans. If we're getting any older than that 50,000 years, then we need to choose a different type of radioactive dating. This is a great video for you to have a look at. I'm going to put this web link for this up on our Compass page in our resources. So this little video here just goes over carbon-14 dating for you in a little bit more detail. It'll explain half-life again to you as well. So have a look when you get it. When we go past that 50,000 year mark that we were talking about with carbon dating, the next big one that we tend to go to is potassium-argon dating. And in this type of decay, it's the potassium which has the 20 protons that's decaying to the argon that only has 18 protons. Now this method and this atom is a lot slower at decaying, so potassium turns into argon at a lot slower rate than carbon turns into nitrogen, which is why we can use the potassium argon dating for things that are older than what we use for carbon dating. So this one here lets us go all the way back to looking at something that's 3 billion years old. So there's a big gap here, and that potassium argon dating is much more user-friendly in terms of fossils that are a bit older. And it is a very common rock that's found, that's found in rocks, which makes it really, really handy. So fossils can take up potassium because we're talking about fossils being rocks. So a great deal of fossils can also have potassium in them as well. Again, this is the one that we use after carbon dating. So if something's shorter in its lifespan, it's been around for not really that long, thinking humans, carbon dating. Anything older than humans, you're going to be looking at potassium dating there. So talking about half-life in a little bit more detail, down the bottom here we have time units. So 1 unit is 1 half-life, and the half-life you'll notice from here, so if we start off with 20 of the original element and then the red one is the daughter element, we get down to here and we've got 10 to 10. So it takes roughly, in this, it's an arbitrary scale of time in this particular scale, on this particular graph. It's not specific. It's not giving us, oh, it takes 50 million years for 50% of them to decay. No, it's different depending on whichever element you're looking at. So when we looked at carbon as the atom, it takes a different amount of time when we compare it to potassium. But 1 half-life is how long it takes for 50% of your parent atoms to turn into your daughter atoms. So 50% of carbon to turn into nitrogen. And then the second half-life is how long it takes for the next 50%, so in this case going from 10 down to 5, and then the next half-life is going from 5 down to 2.5. So each half-life is how long it takes for whatever is there, the half of whatever you're starting with, to turn in to your non-radioactive element that we're looking at. So half-life really depends on the radioactive atom that you're starting with as to how long the half-life will be. Again, in chemistry, you do all the fancy calculations and all that kind of stuff. In bio, all you need to know is that half-life is how long it takes for 50% of the radioactive elements to become non-radioactive. And we can use that to tell how old something is by knowing how long that time scale is. So if we have a time, so say it takes, 20,000 years for 50% of carbon to convert from carbon to nitrogen, then we know that if something has this percentage of nitrogen to carbon, that it's going to be roughly this age. So it's a scale that we're able to use with a lot more accuracy when we compare it to our relative dating that we were talking about not too long ago. Electron spin resonance. This is another type of absolute dating technique, and it's relatively new, but it really depends on the properties that the properties of the electrons that are in the crystal makeup of minerals. So if you think back to year 8, rocks are made of minerals. Minerals have crystal structures. So any type of mineral will have a different type of shape. There's lots of different crystal form shapes that you can be getting. But we're looking at the properties of the electrons that make up the minerals, and the minerals make up the rocks. Very confusing. So crystals in minerals, minerals in rocks, and it's the properties of the electrons in those original crystals and minerals that we are using here in our electron spin resonance. So the minerals collect electrons at a steady rate from their radioactive sources in their surroundings. So they're taking some electrons from radioactive elements that they're surrounded by. So think that carbon-14, for example. They're taking elements from those radioactive sources, and they trap those electrons, and when they're trapped, they become magnetic. So other things are attracted towards them because of that magnetic charge, that north-south magnetism of magnets. We can then use ESR, so the electron spin resonance, to measure the magnetic field that is around the crystal, yeah? So if we can measure how big that field is and how strong the magnetism is, then we know roughly how many radioactive electrons that they've attracted to themselves. So we can use this to date coral, quartz, shells, tooth, enamel, all that stuff is absorbing radioactive material. As it absorbs it, it becomes magnetic. The more magnetic you are, the older you're going to be. So the more time you've had to absorb radioactive materials. If you're not super magnetic, then you're not going to be very old at all. But we look at comparing this with other forms of absolute dating that we get as well. So that carbon dating, radio dating, all that type of stuff as well as the relative comparison dating that we talked about before as well. So electron spin resonance is another one that can come up in your exam. And the last type of absolute dating technique is our luminescence technique. So we've got thermoluminescence, which is the light that's given off by a mineral when it's heated. So if you take the mineral that makes up the rock that you're talking about, or the fossil, you take a small sample of it and you heat it up. When it's heated, you're looking at adding in extra energy, and then the extra energy can then be released as light from those minerals. So we can look at how bright and the type of light and all those types of things that are released, and that's one form. The other one is optically stimulated luminescence. So when it's exposed to visible light, instead of it being heated, it gives off light then as well. So looking at the light that comes from minerals, whether it's heated or put under a visible light source, we can use that to date how old something is as well. So it measures the last time that an object was exposed to sunlight. So if it's quite dull, then it might have been a long time ago. If it's very bright, it's probably going to be more recently. So we can use this one to date things that are from a few hundred to several 100,000 years ago. So it's much more useful than carbon dating, but the go-to one for you guys in terms of your exam, it's more likely going to be carbon dating because there is more research and things around it. But luminescence techniques and the other ones, they're more new. So they can come up in your exam. It's to keep in mind that carbon dating isn't the be-all and end-all. It's not going to be the answer to every question in your exam. If something's older than that 50,000 years that we spoke about before, then you need to look at some of these other techniques. Keep in mind the key information to remember for luminescence techniques. It's light that's given off by minerals that make up fossils or rocks. So it might be a rock that's found near the fossil, and it's the light that the minerals that make up that rock are able to emit when they're heated or when they're exposed to visible light, and that tells us the last time that that rock, mineral, fossil was exposed to sunlight itself. We've spent a fair bit of time now talking about how we know how old these fossils are, but we haven't really spoken about fossils at all. There are a few different types of fossils that you really need to be aware of in terms of what could appear in your exam. The first one is a mold fossil, which is this top picture here. It's where you get grooves and marks left from an organism inside of a rock. The next one is a karst fossil, which is the second photo here. And that's where that gap of where something was gets filled by minerals to make a rock. So they wear away at the bone or the shell, and as the water that's trickling through these rocks wears it away, you get left with the mold. That mold spot then gets filled, or that hole gets filled with the minerals that are brought through by the water that's trickling through these layers, and it gets deposited inside, and that's where you get the shape of the mold. The next one is trace fossils. Now these are fossils that show life, like the footprint down here, but they aren't an organism itself per se. So it's your footprints, nests, burrows, anything that shows life but isn't life itself. And then you've also got true form fossils. Sometimes these are referred to as body fossils, where you get the whole organism. So this one down here is a little lizard that's been stuck in tree sap or as amber. This is the one that you might recognize if you've watched the original Jurassic Park. The originals are always the best, remember? And the little mosquito gets stuck in the tree sap, and then they get the DNA out of the mosquito. That's what that type of fossil is there. So it's a true form or a body fossil, where you get the actual animal or the actual part of an animal that makes up that type of fossil there. So we looked at types of fossils, and we know how to date fossils, but how do we actually get fossils? It's a thin tissue. There's a few different things to think about. It's the bones, and it can be the footprints. So there's a whole different range of ways that things can be fossilized. If you're looking at whole body fossils, there are some great YouTube clips around how these fossils are made. Generally, they die near a water source or a tar pit or something that's able to quickly cover them and have them removed from oxygen so that they're not eaten by animals and pulled apart and all those types of things. The other ways that you can get fossils are by freezing them. So think like woolly mammoths in polar ice caps and all that type of stuff. And if things dehydrate, meaning that they don't get enough water, so they dry out really quickly. Those types of ones are very common in deserts. But we're looking at creating an atmosphere where the organic matter that makes them up doesn't decay very quickly. So anything that's super cold, anytime it can't get oxygen, all those types of things are going to support the organism to not decay, but get covered by layers of rock or sand, so that over time more and more layers build up and it pushes down on the organism. Water will trickle through and wear away at whatever was in there. So that's generally roughly how fossils are formed. They need to die somewhere where they're going to be able to be kept safe. That has a good amount of water source so that layers can build up on top of them, and so that minerals can be brought through the layers of rock to wear away at the organism, such as its bones or its shell, and then you can fill those holes to make your mold and cast fossils and those types of things. It's also important to know that plants can be fossilized, and the name of fossilized plants is petrification. So you might have heard of petrified wood. In this case, the plant tissues are replaced with dissolved salts that are in the water, and over time those salts then turn into rock. So petrified plants are plants that have become rock. They are fossilized plant material. So step by step how we get fossils. Number one, the organism needs to die to start with. It's not going to survive this process. It's becoming a rock, so it's going to have to die. Where it dies is quite important. If it dies near water, so seas, lakes and rivers, that's very handy. It's going to get covered by water, and that's going to stop the decay process quite nicely. If it's on land, it needs to be covered by sand or volcanic ash, or get stuck in something like tree sap or tar or in ice. All of those things need to happen, or one of those things needs to happen, so that it doesn't decay and so that it's able to be slowly worn away and broken down by the minerals. So that's step number one. It has to be covered by something. The next part that we're looking at is the chemical changes that occur. So we're looking at the petrification that we just spoke about with plants. So progressively replacing the hard parts of the original organism with minerals like quartz or opal, so that as it gets worn away, it gets replaced by a mineral, and gradually it's going to become a rock over time. Now, the other option to it being replaced over time is it being completely dissolved away. So this is where you get your mold, and then when it fills up again, it's going to become a cast. So the organism itself is stuck in a layer of rock, and it's going to be worn away by water and other things that are going through the gaps in the layers of rock. When it's worn away, that can create the mold fossil that we were talking about before. And then that cavity can get filled by other minerals, and then that becomes the cast fossil that we were just discussing as well. Step number 4 is after its burial, it needs to undergo a number of chemical and physical changes depending on the environment in which it's enclosed. So this is looking at creating sedimentary rock. There needs to be a decent amount of pressure pushing the layers of little rock together around the organism so that the layer of rock forms around whatever has died and been buried. And then you need to get successive layers of rock, so many more layers of rock building up above the one that has just formed where your organism was, so that it is protected from the environment around it, so it doesn't erode, doesn't wither away, it's able to be kept relatively safe. Now, as those layers build up, it's pushing down quite hard, so you're looking at having a decent amount of pressure on this layer of rock, so it's getting squished smaller and smaller, so it's going to get flattened out a little bit, and when it gets flattened out, that's going to cause some distortion. So it's not going to look exactly the same as what it looks before. It's kind of like a Mars bar and you squish the Mars bar. You can tell it's a Mars bar, but it's not the same as what it was before. Let's be honest. It's all squished. Everything's a bit flatter. And then once that happens, you're also looking at heat and pressure acting on those rocks to help them form and stay the way that they are. So the rock forms need to be occurring. And to get the rock that you're talking about being your fossil, you need a decent amount of heat and a decent amount of pressure. So what we have here is a nice little example around a hominid and how it has become fossilized. So in this particular example here, our little hominid has died close to a water source, maybe beside a river. And when it's died, it's been nice and close, so the soft tissues decay away, but the bone's been able to be left. Okay, so it hasn't been eaten by an animal and dragged away. Okay, it's died near the water source. Water levels for this river or lake rise and they cover the hominid. Weathering, erosion bring lots of sediment into this, and the sediment starts to build up on top of the fossil bones, the bones that are going to become the fossil. Okay, and the same thing's happening to the footprint. So the footprint was there, it's dried out, and then layers of other rock are starting to build up on top of it. Now, some of the examples that they have in the text to the left here around how these layers are forming is it can be a volcanic eruption. So ash from a volcanic eruption, not the lava because that's going to kill everything, but the ash that comes out of the volcano slowly filters down, and it's quite cool, and that can build a layer over the top of it as well. Over time, the area will dry out. It might go through a drought, and then the layers start to become exposed again. So erosion of the different layers of rock starts to occur so that people are then able to find the hominid bones, and then it gets studied to look at the fossilized remains. So we're looking at tools that they might have used, marks, how it died, all that type of information that we can gather from this fossil. So this is a rough little how a fossil gets made picture here for you, which is a great little summary again. There are some great YouTube clips on how fossils were made. I really recommend that you have a little Google and have a little watch of some of them. You don't need a great deal of detail here. It's more around things that impact how fossils are made, just so you have a good general understanding about the factors that are impacting on fossils, so why they are so rare and all that type of stuff as well. Again, 50 million fantastic YouTube videos for you to have a look at there. Biogeography. So this one here comes from the study design as well. It was one tiny little word in our dot point, but biogeography is the study of the distribution of species and ecosystems in geographic space and throughout geological time. So it's where plants and animals and ecosystems were. Remembering ecosystems are the bigger picture of everything altogether. So your plants and how they interact with animals and all that kind of stuff. So it's where they were in geographic space, which means where they were on planet Earth, which countries they were in, which tectonic plates they were on, all of that kind of stuff, and how they've moved through geological time. So we're looking at that history of the Earth again. So biogeography is where things lived over time and what they also lived with. Now we can track different fossils and their different areas as well as how old they are quite well. So we can look at a fossil remain in one particular area, say over here, and we can find very similar fossils over here in Africa, which tell us that at a period of time, South America and Africa had to have been joined together, and these reptiles, which were roughly 3 meters long, must have been able to very easily get from one area to the other. So that's what biogeography maps tell us. What land was connected? How did things move from one place to the other? How do we know that land was connected to each other? And what were their ecosystems like? So how did they get around? Was there a lot of water? All that type of stuff. We get that from biogeography. Developmental biology is the next one that we have here, and we're looking at it's the study of how plants and animals grow and develop, and we can look at comparing their embryos and how they've developed over time. So this bottom picture here is humans and how humans have been evolving, but if we look here at these middle two here, they look pretty similar. This bottom one down here is a chicken, and this one smack bang in the middle is a tortoise. Now they're very similar in terms of their embryo shape, but we know that a chicken is definitely not a tortoise, and a tortoise is definitely not a chicken. They're two different animals, and yet their embryos look so similar. And we can say the same thing up the top here with the fish and the salamander. While they're different organisms, their developmental biology in terms of going from young embryos to life is very similar. The thing that we can look at comparing in terms of determining how related things are and how things have changed over time is structures that are found within different organisms. So they're structural morphology. So morphology is a branch of biology, and we're looking at the structures and the specific features of different organisms, okay? What we're going to be talking about here is homologous and analogous structures, so ones that show that they had a common ancestor and then they've become different organisms, versus things that have similar features but don't actually have similar bone structures and limbs that show that they were relatives of each other, but maybe they're two entirely different things that just happen to show similar characteristics. So we're looking here generally at outward appearance, so shape, structure, color patterns, all those types of things. That's external morphology. When we look at bones and organs and how things function, that's when we're looking at internal morphology. So the examples that we have here are a turtle, so it's the fin of a turtle, of a dolphin, the forearm of a human, and the leg of a horse. Now all these things contain these bones here. They vary in their length, but they are all seen in these 4 different organisms, including on the side here the fruit bat and the bird or the chicken example that's provided. So what that shows us is that at some stage these things were probably related, which is why they have very similar bone structures, but different selection pressures, different things have happened to them, so they've all become different organisms, they become different animals. But we're looking at those similarities in their bone structure to show relatedness at some point in time. So again, these homologous structures are ones that are derived from a common ancestor. So they share a plant or an animal in their past, and then they've diverged away from each other to become less similar to each other. So we're looking at different leaf shapes and all those types of things. So the examples that we have here are the pitcher plant, which is the top one here, and this one here is our Venus flytrap. Both of these plants are able to break down and digest insects and get nutrients from them. So that's a homologous thing that they're both able to do. We're also looking at these bright red leaves. Now these are leaves and not flowers. And then we're looking at the stems on a cactus and how they've changed their leaves and things like that, so that they're protected from water loss and plant eaters and all those types of stuff. So we can look at these characteristics and where they're commonly seen in other plants to determine how related these organisms are to each other. Is it that they've just got something similar to each other but they don't share a common ancestor? But when we're looking at homologous structures, the key to having a homologous structure is that they have to have that common ancestor with each other, okay? So these guys here all share common ancestors. When we look at analogous structures, these are ones where they don't share a common ancestor to each other. For example, a bat wing versus an insect wing. They have a similar function. They both help things fly, but they have very significant structural differences. So the bone limbs that make up the bat wing are very different to the ones that make up the insect wing. Now when we look here at the bat and the bird, these ones are homologous because they share those common bone structures versus the insect wing, which doesn't share the common structures. Homologous, common ancestor, similar structures. analogous, no similar structures, just they do roughly the same function, but they don't have a common ancestor that they've evolved this skill, attribute, structure, whatever it is from, okay? There are two patterns of evolution that you need to be aware of. So when we're talking about that homologous structure, sharing of common ancestors versus analogous structure, no common ancestors, and they are divergent evolution and convergent evolution. In divergent evolution, they share this common ancestor and then they've changed over time. So think about the last topic that we were covering where we're looking at natural selection, environmental pressures, all those types of things create change. Slow, gradual, consistent change over time that creates all new species. So that's what's happening here. So these guys here all share a common ancestor, but over time their selection pressures have made them all different to each other. And this is what we also refer to as adaptive radiation. So we have a variety of species that have evolved from from a single ancestral species. So if you think about Darwin's finches, which we're going to talk about in a little bit, they have one common finch ancestor that they've all diverged from. So divergent is when you start as one and then you become many different types. So it's a response to selection pressures, as is convergent evolution. These guys here, so the falcon, the bat, and the pterodactyl that we have on the end here, don't actually share common ancestors, but they all have wings. Now this is adaptive convergence. All three of these animals have developed wings to help them do better and succeed and stay alive in their natural habitat, but none of them share a common ancestor. So genetically, they aren't related to each other, they don't have similar structures to each other, and yet they're all able to fly. So there's superficial similarities. They have things that do the same thing. So in this case, they all have wings, they all help them fly, but they are not actually in any way related to each other. So when we're talking about divergent evolution, it's that common ancestor making two or more new species. And so they're going to be having those homologous structures that we spoke about before. If you have a common ancestor and you've just evolved slowly and gradually away from that common ancestor, then you're going to be sharing characteristics with other organisms that also evolved from that common ancestor. So an example to think about here is the camel and how camels have evolved. So they've looked at the different versions of camel over time, as you can see up here. And if you look down here at the 54 million years ago, this guy looks pretty similar to some animals that we might be seeing out and about, like our deers and our fawns and all those types of things. So that's some superficial in terms of how it looks, but then they will dive in deeper and they'll compare the bone structures, so the limb bones and all those types of things as well, and those that have been fossilized, so they can track the evolution of the camel over time, how it's changed, what are the selection pressures that have created this change, and all those types of things as well. So this is an example of camels changing from their ancestors into the way that we see them today, and there will be other very similar animals that diverged from the common ancestor of the camel as well. Another one to be looking at in terms of adaptive radiation and that change over time, becoming lots of different species, the prime example is Darwin's finches. So on the Galapagos Islands, there's a whole heap of different finches on each island, and each population of the finches has a different food source that it primarily relies on. So this guy at the top is leaves, this guy's is buds and fruits, and we've got grubs. Based on the food that they're eating, you'll notice that their beak types are different. Now they are all finches, but they have adapted, they have changed to suit their environment and the foods that they are eating. Now we spoke about this with natural selection, and adaptive radiation sits within there as well. So they have one common ancestor, this guy in the middle who eats seeds, but based on the selection pressures in their different environments that these other finches live in, they've all developed slightly different beaks so that they're able to feed, live, survive, be happy and healthy in their new habitats that they live in compared to the guy that eats seeds. So natural selection can be something that causes divergent, like the form of divergence that is happening here when we talk about divergent evolution versus convergent evolution. So one common ancestor, selection pressures that are different on the different finches creates all the different bird beaks that you see here. This here just provides you with a little bit more information about Darwin's finches, so feel free to hit pause and have a good read. The key things to know is this is just something that is proposed by Charles Darwin, and it is a very classical example of natural selection or allopatric speciation, so creating new species, and divergent evolution here as well. As I mentioned before, we're looking at the Galapagos Islands, which are out in the Pacific Ocean. If you are looking for some more information, there's a whole heap of really, really awesome stuff again on YouTube to be looking at. Have a look at this web link down here. That might be a good place for you to start. But we're looking at how one species of finch looks very different in some ways, but also very similar in other ways to the finches that they're seeing on the different islands, and how they have become different species over time based on their particular food type, creating that selection pressure for these finches to change within the environment that we're finding them. Convergent evolution, so let's dive into this one here a little bit more. Now, remembering these are no common ancestors, so it's not due to a common ancestor, but they may have homologous structures, so structures that are similar, or they might be able to do a similar job, so think the wings and all that type of stuff, but they are unrelated in terms of their taxonomy. Now, there are a few different examples here on the right for you to have a look at. So the niche is the type of environment and or the things that these things do and how we're going to categorize them. So the top one that we have is things that like to burrow. So we have a mole and we have a marsupial mole. Now, what we're comparing here is placental mammals to Australian marsupials. Now, mammals and marsupials are two different things. If you're not sure of it, I really recommend that you have a little Google and a read before we go into these too much deeper. The next examples down are an anteater and a numbat, and you'll notice that the numbat has anteater in brackets here, but they're not actually related to each other. We have a mouse and we have a marsupial mouse. Again, very similar animals, but they don't share a common ancestor. Even down here, when we look at things that glide, so our flying squirrel versus our flying flanger, that's a hard one to say, those guys aren't related to each other. They just share common traits. They might have common structures to them as well. They might be able to do similar things, but they are definitely not related to each other. And the next little bit that we're going to be covering, in fact, the last little bit of this study design section for you, is mass extinction. Now, it's important to know that it can have many different causes in terms of things reaching the end of their sustainability lifespan. So the example that we have here, you may or may not have heard of this lovely bird. It is the dodo bird, which doesn't exist anymore. But your other examples for mass extinction are things like dinosaurs. And what we're talking about here is a complete loss of habitat, so that the organisms are not able to live within there. It can be caused from competition, so an entire organism or species can go extinct because of competition with other animals, so that they're fighting for their food sources, and maybe one animal doesn't get it, but the other one does, so one of them dies off because it doesn't get any food. That's the example of competition extinction. There's lots of different forms of extinction. There's lots of different ways that it can happen. Us humans, we have an impact on this as well. So by hunting things, so think about the white rhinos and hunting for their tusks. Animals and plants go extinct all the time, and it can be due to how they survive in our climate, how much oxygen and carbon dioxide and nitrogen they're able to tolerate in the air. There's a lot of different things that can cause extinction in general. But when we're talking about mass extinction, it's that global catastrophe. It's not the once-off hunting of an animal so that it doesn't exist anymore. Mass extinctions go beyond that. We're talking about a whole heap of species going extinct at once. And the big example that you're probably all thinking of right now inside your head is the asteroid impact that they are proposing caused the extinction of the dinosaurs. So there's evidence to say that there was an asteroid impact, and that was believed to be the cause of the mass extinction, that 70% of marine and terrestrial, so when we say terrestrial here, we're talking about land-based animals. So 65 million years ago, an asteroid hit planet Earth, and it completely changed how it is. So it can cause the ice age, it changed climate and all that type of stuff. And it's believed that that is the cause of 70% of the death of different species. And so then we're looking at evolution that's occurred from the surviving ones beyond that. So mass extinction is it's not just one species, it's a whole heap of stuff dying off at once. And this video here is another good one for you to have a look at, so I'll chuck the web link up for you in our Compass resources for you to have a look at when you get the time. Thanks for watching today, guys. I hope this helps you to prepare in terms of your exam and your SACs and everything that you've got coming up. Make sure that you get your study notes written. I hope you hit pause a whole heap because this is a very long video at 55 minutes. But I hope that you've got everything that you need to be successful in this topic. I'll see you guys later.